Now, the last thing I want to share to you is all about goods buffers. So this is a set of buffers commonly used in biochemical experiments developed by Norman Good, and these are organic molecules. Now, they are very advantageous over our inorganic weak acids and their conjugate bases because, well, for one, all of them, most of them have a pKa between 6 and 8. And we want that, right? Because any buffer whose pKa is between 6 and 8 means that it can maintain the pH at the physiological level. Next, these molecules have very high water solubility. So we have some of our inorganic weak acids that are quite difficult to dissolve in water. So maybe some of them needs a little bit of heating so that they can completely dissolve in water. Some of them needs a lot of stirring, but these goods buffers dissolve in water easily, which means their preparation is very convenient. And we are sure that all of the components are dissolved in our solution. Next, they have low cell membrane permeability. So this characteristic is important if we know that our reaction is something that is taking place inside the cell. And if you are performing your biochemical experiment in an environment that is mimicking the inside of a cell, then a buffer that has low cell membrane permeability is good because it stays inside your cell mimic. They also have minimal salt effects because in the first place, they are organic molecules, unlike our inorganic weak acids and their conjugate bases. If you notice, most of the time, the conjugate bases come from salts. So you have some floating ions in solution that are not really participating in the buffering action, but they are in fact, they have an effect. So we call that salt effect. Next, these organic biological buffers have consistent acid-base dissociation constants, the Ka values. So you should know that your Ka values change when temperature changes. So they are temperature dependent. So most of our weak acids, their Ka values change with temperature and actually even with concentration. But with your goods buffers, their Ka values change very, very slightly with a change in temperature or a change in their concentration in solution. So we want that, right? Because our computations are more accurate. Next, these buffers have minimal interactions between buffer components and critical reaction components. So an example would be metals. So for example, your biochemical experiment needs some metals like magnesium ions. So these buffers do not interact with such. Okay, so we know that some ions, they can chelate your metals or your cationic metals. And we don't want that because the metals are important in the biochemical reaction. And of course, these buffers have high chemical stability. So many of them can be kept in the shelf for a long period of time, unlike some of your inorganic buffers. They also have low absorption spectra in UV and visible regions, and this is important if we are quantifying your biochemical sample using this technique, spectroscopy. So it's good because if your molecule biomolecule is the one absorbing and it is in the environment of the buffer, since your buffer is not absorbing, then any absorbance that you measure will only come from your biomolecule. And lastly, they are easy to use. So an example of a goods buffer is the MES buffer. So MES is short for this name of the molecule, 2N morpholino ethane acid. So you have this ring structure, so that's where you get the morpholino, and then you have the ethane sulfonic part. Now, how does this act as a buffer? We only see one molecule. Where are the two components? So that this is what's unique about your goods buffers. You have one molecule. This molecule will act as your two components. So how does it happen? So if we dissolve this molecule in solution, its form is not like this. So what you will see is the nitrogen in the ring has a proton attached to it. So why is that? It's because this nitrogen has a lone pair. So that lone pair can accept a proton. So you have a proton attached to this nitrogen, and the hydrogen of the sulfonic acid group is removed. So this is actually just O negative. Do you see how this can act as the two components? So in this form of the molecule, if you add an acid, you have this O negative to neutralize the added acid. And if you have a base, you have this protonated nitrogen to donate a proton to neutralize the base. So that's how, or this is an example of the mechanism by which your goods buffers work.